Hello everyone, I'm Anqing Hou, a postdoc in Professor Yiqi Luo's lab in North Arizona University. Today I will talk about matrix phosphorus model and data assimilation. This talk is related to the chapter 3.3 in our land carbon cycle modeling book. This talk is mainly based on our paper published on GGR by Geoscience in 2019. The study used the key modeling skills you are learning during this training course. They are matrix model and data assimilation. If you are not familiar with phosphorus, you may wonder why are we concerned about phosphorus. The reason is that all life forms need phosphorus for growth and reproduction. Almost all life forms use DNA or RNA as the carrier of genetic information, and use ATP as the carrier of energy during biochemical reactions. Phosphorus is a key element of these macromolecules. Our recent study showed pervasive phosphorus limitation of plant production in terrestrial ecosystems. Similarly, another study found that soil phosphorus availability can limit the response of plant growth to CO2 enrichment. Phosphorus for plant growth in terrestrial ecosystems derives mainly from soil. Given the important role of soil phosphorus supply in plant growth, primary production, and terrestrial carbon sequestration, more and more land models are incorporating soil phosphorus dynamics, including Cable, CLM, OKD, and some other models, to constrain model predictions of land carbon sequestration. Currently, we know that soil phosphorus dynamics regulate soil phosphorus supply. We also know the main processes of soil phosphorus dynamics, but we lack an appropriate way to quantify these processes. Previous studies used radioactive isotopes and expensive equipment such as an isotope ratio mass spectrometer to quantify soil phosphorus dynamics. Soil phosphorus dynamics is difficult to be quantified using experimental approaches. Data assimilation has the potential to quantify ecosystem processes that are difficult to be quantified experimentally. So we were wondering, can we use data assimilation to quantify soil phosphorus dynamics? This talk will tell you that the answer is yes, because like terrestrial carbon dynamics, soil phosphorus dynamics is also composed of pools and fluxes. In this study, we constructed a soil phosphorus model with matrix equations and performed data assimilation analysis of soil phosphorus dynamics by combining the soil phosphorus model with measurements of Hadley soil phosphorus fractions. The purpose was to quantify soil phosphorus dynamics. The data used in our study was from Guo and co-authors 2000, published on the journal Soil Science Society of America journal. Guo and co-authors reported 8 wonderful datasets of soil phosphorus fractions, including 3 datasets in slightly weathered soils, and 5 datasets in strongly weathered soils. In general, they used crops to remove labile phosphorus from those soils to trigger changes in phosphorus fraction in these soils over a total of 14 cropping periods. After every 2 croppings, they sampled a small amount of soils to determine soil phosphorus fractions. They planted crops in plastic pots in a greenhouse and determined soil phosphorus fractions using a commonly used soil phosphorus fractionation procedure, that is, the Hadley soil phosphorus fractionation procedure. Here's a summary of the basic physiochemical properties of the 8 soils. As you can see, these 3 soils are the slightly weathered soils, and the other 5 soils are the strongly weighted soils. The slightly weighted soils have relatively high total phosphorus concentration, high soil pH, but relatively low soil organic carbon concentrations. Here shows one of the 8 datasets. Soil phosphorus fractions were grouped and classified into 5 soil phosphorus pools. They are labile phosphorus, which is readily available for plant growth; organic phosphorus, which is phosphorus in organic form; secondary mineral phosphorus, which is phosphorus associated with secondary minerals such as aluminum oxides and iron oxides; primary mineral phosphorus, which exists in apatite; and occluded phosphorus, which is thought to be in occluded forms and unavailable for plant growth. Here shows our soil phosphorus dynamical model constructed for the dataset. The model considered the key ecosystem phosphorus processes, which are plant uptake, as shown here, soil weathering, as shown here, mineralization of organic phosphorus, as shown here, microbial assimilation of inorganic phosphorus, as shown here, and also desorption, dissolution, sorption, and precipitation processes, and also including the solid phase transformations of phosphorus. These processes can be mathematically described by the 6 equations here, or by the 1 matrix equation. Clearly, the matrix equation is simpler in form and also in coding. The matrix model is also much easier to understand. Here dt indicates time interval or time step, which is day in our model. dBT, dPt is the change in soil phosphorus pool. In other words, it's the difference in soil pool size between 2 times. A is the coefficient matrix, which describes the transfer rate of phosphorus among soil phosphorus pools. For example, A12 indicates the transfer coefficient of phosphorus from pool 2 to pool 1. -1 in the diagonal cells indicates the transfer out of phosphorus from the pool. K is the turnover rate matrix. For example, K1 indicates the turnover rate of labile phosphorus. K2 indicates the turnover rate of soil organic phosphorus. PT indicates the size of each soil phosphorus pool at time t. The data assimilation system we used here is the same as you will learn in the coming units 6 to 10. In simple, it used the Bayes' theorem to optimize parameter values. Now let's look at the results. As you can see, posterior distribution of the turnover rate of soil phosphorus pools were constrained well by the datasets. Here's the example of one of the 8 soils. Here shows a comparison between simulated soil phosphorus pools and measured soil phosphorus pools. Here, the open symbols indicate simulated soil phosphorus pools, and solid symbols indicates measured soil phosphorus pools. As you can see, simulated soil phosphorus pools after decimation matched well with the measured soil phosphorus pools in all soils and in all pools. And here shows the optimized parameter values. And you can see optimized parameter values varied essentially among soils. For example, the turnover rate of label phosphorus varied about threefold across 8 soils. The turnover rate of soil organic phosphorus varied over fourfold across soils. Here we show the bivariate relationships among parameters, which provide insights into the relationships among soil phosphorus processes, like the relationship between K1 and K2 indicates the association between the turnover of a soil label pool, label phosphorus pool, and soil organic phosphorus pool. Our results also enabled an exploration of the sensitivity of soil phosphorus pools to soil phosphorus processes. For example, the pool size of soil label phosphorus was mainly determined by its turnover rate, K1. Finally, our results enabled a quantitative study of environmental controls over soil phosphorus dynamics. By linking the estimated parameter values with soil physiochemical properties, we found some significant relationships between soil phosphorus dynamics and soil pH and organic carbon concentration. For example, we found that secondary mineral phosphorus turnover faster in neutral soils than in acidic soils, and turnover faster in organic carbon depleted soils than in organic carbon enriched soils. Not all label phosphorus is directly available to plants. As you can see, the proportion of label phosphorus to plants is varied largely from less than 0.1 to about 0.6. The variability of label phosphorus to plants is higher in neutral soils than in acidic soils, and is high in organic matter enriched soils than in organic carbon depleted soils. In summary, we performed a decimation analysis of soil phosphorus dynamics with the matrix phosphorus model and provided new insights into soil phosphorus dynamics and availability. This helps a realistic model projection of soil phosphorus dynamics and availability, which will finally aid our management of phosphorus resources for key ecosystem services such as land carbon sequestration and food production. Finally, I want to thank my co-authors for their contribution to this work and the financial support of this study from South China Botanical Garden of the Chinese Academy of Science, Northern Arizona University, National Science Foundation of China, and the National Science Foundation of USA. Thank you all for your attention. That's all for this talk.